This slide presentation is part 1 of 2 of the lymphatic system and the immune system. We're going to start by looking at the anatomy of the lymphatic and immune systems. And the lymphatic system is comprised of specialized vessels, cells, and organs that carry excess fluids that came from the bloodstream initially and went into surrounding tissues back to the bloodstream. And as they carry that fluid back to the bloodstream, they go through special organs that are designed to help filter pathogens and antigens. And antigens are things like bacteria or bacterial toxins, viruses, cancerous cells, incompatible blood cells. Anything that can elicit or provoke an immune response is an antigen. When fluid enters into lymphatic vessels, into the lymphatic system, it's known as lymph or lymphatic fluid. And one of the things that the lymphatic fluid will filter through are specialized organs known as lymph nodes. Lymph nodes are small, they're bean-shaped, and they serve as a staging area for the development of an immune response. When fluid moves from arterioles and capillaries into the surrounding tissues, it's known as interstitial fluid. Interstitial fluid can then enter lymphatic capillaries. Those are the smallest lymphatic vessels. Lymphatic capillaries are blind-ended vessels. They kind of look like the end of a tube. And so the lymphatic capillaries have cells that overlap one another, and as interstitial fluid enters into the area in the tissues, as blood pressure forces some of the fluid out of the blood vessels into the interstitium fluid enters those lymphatic capillaries, and the overlap serves as a one-way valve that's anchored by collagen. Now we have lymphatic capillaries and lymphatic vessels interspersed throughout areas where we have arterioles and venules and capillary beds. However, there are some places where we don't find lymphatic capillaries. So those would include the central nervous system, bone marrow, bones, teeth, as well as the cornea of the eye. And again, when interstitial fluid is created, it's picked up into the lymphatic capillaries because of that overlapping cellular construction. There are specialized lymphatic capillaries found in the intestines. Those are called lacteals. Lacteals carry lipids and lipid-soluble particles into the bloodstream. When the fluid enters lacteals, because the lymphatic fluid in lacteals contains triglycerides and other lipids and proteins that will be carried to the liver and then ultimately the bloodstream, that milky fluid in lacteals has a special name, and that's referred to as chyle, C-H-Y-L-E. If we look at this particular picture, you'll notice that there is a blood capillary network surrounding a lacteal, which is a closed-end capillary, lymphatic capillary, and that's seen in this villus. There is an intestinal villus, which is a finger-like projection, and you can see that in B in this picture. And you'll also notice there's a crypt, an area where between the intestinal villi. And so there's a Lieberkuhn crypt in this picture. I know we haven't done the digestive system yet, but I just wanted to point out those structures. So as molecules are broken down in the digestive system, the lacteal is designed to absorb those lipid and lipid-soluble particles. Lymphatic capillaries converge into lymphatic vessels. Lymphatic vessels have one-way valves that keep the lymphatic fluid moving unidirectionally. Those lymphatic vessels will further merge into larger lymphatic trunks, and the lymphatic trunks will finally merge into structures known as lymphatic ducts. At the upper end of this picture, on the left side of the picture, you'll notice the right lymphatic duct that's going to enter into the right subclavian vein close to where the internal jugular vein is. And we also see the thoracic duct on the left side of the person, which is the right side of the picture. At the top, you'll see it says entrance of thoracic duct into the subclavian vein, again close to where the internal jugular vein enters. So lymphatic trunks merge into lymphatic ducts, which are going to dump into the bloodstream. So we have interstitial fluid that forms from tiny blood vessels as blood pressure forces fluid into the interstitium. That gets picked up by lymphatic capillaries and then eventually dumps back into the bloodstream. Now you'll notice in this picture there's also a special structure called the cisterna chyli. It's a sac-like chamber that receives lymphatic fluid from the lower abdomen as well as the pelvis and the lower limbs. In this picture we also get to see lymph nodes that are located in specific locations throughout the body. And where there are lymph nodes, there tend to be kind of a little conglomeration of lymph nodes. Also, on the left side of the picture, you'll notice about midway it says there's an aggregated lymphoid nodules being presented within the intestinal wall known as Peyer's patches. So we have lymphatic tissue in the wall of the intestines. Again, that's called Peyer's patches. When it comes to immune responses, we have two different types of immune responses. We have innate immunity and adaptive immunity. Those are the two main branches of our defense system in our body. However, they differ in terms of how quickly they can respond, how specific the response is, and how they work in terms of the mechanism of action in response. Innate immunity is very rapid, it's non-specific, and it's there from birth. Whereas adaptive immunity it's a slower response, highly specific, and develops over time. Now, when we look at our immune response, there are three temporal phases of immunity. We have recognition, response, and resolution. And when it comes to innate immunity, that with which we are born, we have barrier defenses, which include our skin. Our skin forms a barrier against different types of pathogens, and that includes the keratin in the skin. The skin is also acidic, which can help kill certain bacteria. We also have our mucous membranes and gastric secretions. Again, those are all innate. We're born with them, and they're involved in primary defense because that's our first line of defense. However, our primary defense system also includes a first encounter with an antigen. Antigens are things that evoke an immune response, and again can be things like bacteria, toxins, cancer cells, viruses, even incompatible blood cells. So anything that provokes an immune response is an antigen. So we have a primary defense system against antigens that includes special leukocytes known as lymphocytes. Lymphocytes come in different flavors. We have B cells and T cells. So if we look at this picture, we can see that with hematopoiesis, which is the creation of blood cells, we have myeloid stem cells and lymphoid stem cells. Myeloid stem cells differentiate, and we've seen this with blood cells. Remember, we get megakaryocytes, which turn into platelets. We get erythrocytes, and then we get certain white blood cells known as leukocytes, including granular cells, the basophils, neutrophils, eosinophils, and we also have agranular cells, which are monocytes and lymphocytes. So monocytes turn into macrophages, which are big eaters, and then the lymphoid stem cells will differentiate into lymphoblasts, and from there we see natural killer cells and T lymphocytes and B lymphocytes. B lymphocytes can differentiate into plasma cells. Now we're going to take a look at how these cells work, and I want you to understand that with innate immunity, as far as cells go, we have specialized cells, soluble factors, and these are non-specific. And part of that innate immunity, that primary defense mechanism, includes what we call naive B cells and T cells. Naive B cells and T cells are part of innate immunity. But then we also have adaptive immunity, and our lymphocytes are really important in controlling our immune response. And adaptive immunity is really, it's where we have specificity to certain pathogens or antigens. So there are three kinds of cells involved in secondary defense, which is adaptive immunity, and those are phagocytic cells, lymphocytes, and granular cells. In looking more closely at our primary response versus our secondary response, let's look at the primary response where we said we have specific lymphocytes known as naive B cells and T cells. Those have not yet encountered a specific antigen, and those are going to be initially involved in a primary response. So when we have that initial exposure to an antigen, the naive B cells are going to become active, and they will differentiate into plasma cells. Now, the thing that's important to understand about plasma cells is that those are the cells that produce antibodies. Antibodies fight antigens. However, upon initial exposure to an antigen, the naive B cells will differentiate into what we call memory B cells. The T cells will differentiate into what we call memory T cells. Now, the great thing about memory cells is that these are long-lived cells that retain immunologic memory. So they'll remember how to respond to the antigen if they come across it again. And that's when we have a secondary immune response. Memory B cells and memory T cells that were generated in that initial primary response can now be rapidly activated upon re-exposure to that same antigen that they saw in the primary response. And memory cells can very quickly differentiate into plasma cells, able to produce even more antibodies, and cytotoxic T cells that can kill infected cells. That leads to a faster, stronger response compared to the primary response. So while both responses are going to fundamentally rely on those two classes of adaptive immune cells, the B and T lymphocytes, it's the state of differentiation and the involvement of the memory cells in the secondary response that's going to cause a more efficient, more effective immune response when a second or re-exposure to that same antigen occurs. We've looked at the first three types of lymphocytes on this chart. However, we didn't mention yet natural killer cells. Natural killer cells are special cells that are part of the innate immune response, and they're designed to go after virus-infected or cancerous cells. Next, we're going to look at primary lymphoid organs, and this is where lymphocytes proliferate, where they mature, and where they're selected to fight against a pathogen or antigen. Now, when we look at bone marrow, and you'll recall from AMP1 that bone marrow is made from reticular connective tissue, we have two types of bone marrow. We have red bone marrow and yellow bone marrow. Yellow bone marrow is yellow because we have more adipose connective tissue mixed in with the reticular connective tissue, and it has a yellow appearance. And that's because of the adipose having that appearance, and that's where we store energy as fat. However, red bone marrow is where hematopoiesis, the creation of blood cells, occur. So B cells and T cells develop in red bone marrow. And the B cells will develop there and become mature. However, the T cells migrate away from the bone marrow to the thymus, which, by the way, is most active during childhood. And that's where they're called thymocytes. Once they get to the thymus, they're referred to as thymocytes. And that's where they will become more mature. And the thymus is an organ that is found between the sternum and the aorta. You can see it in the picture above. It is going to be largest during childhood, where it's most active, and then it decreases in size and actually stops functioning during adolescence. Next, we're going to take a look at secondary lymphoid organs and the active immune response. Secondary lymphoid organs include organs like the lymph nodes, spleen, and tonsils, where naive lymphocytes are going to encounter antigens, become activated, and initiate an immune response. Secondary lymphoid organs have the following tissue characteristics. They have lymphoid follicles where lymphocytes are formed, and they have specific B cell and T cell rich areas. So there's a lot of T and B cell cells in those places. They also have a structure of reticular fibers, because remember that it's reticular connective tissue, that have associated macrophages that are fixed onto the fibers. They include germinal centers, which are the sites of rapidly dividing and differentiating B lymphocytes, and they also have specialized post-capillary vessels known as high endothelial venules. And the cells that line those venules are thicker and more column-shaped than normal endothelial cells, which allows cells from blood vessels to directly enter those tissues. When we look at the structure of lymph nodes, we know that lymph is going to flow through lymphatic capillaries and vessels and then through lymph nodes on its way back to the bloodstream. And lymph will enter the lymph node. On the left side of this picture above me, you'll notice that there are afferent lymphatic vessels that will bring lymph fluid into the lymph node. The lymph node is surrounded by a connective tissue capsule that invaginates into the node, forming a network of a lace-like or lattice-like pattern known as a trabecula. And there are also special spaces called sinuses through which the lymph will flow. There's a subcapsular sinus and a medullary sinus, and those sinuses are going to be the spaces where the fluid flows before it exits the lymph node through efferent lymphatic vessels. Now, you'll notice that the lymph enters the node on the convex side and exits the node on the concave side, where you see the term hilus. So the hilus or hilum is that indented portion of the lymph node where the fluid will exit. You'll also notice that there are follicles in the lymph node. the follicle is the darker purple area, and the lymphatic follicle, having a problem speaking, or lymphoid follicle is going to consist of really tightly packed lymphoid cells. And then the follicles inside often have lighter staining germinal centers. And in the germinal centers, that's where proliferating B cells are going to predominate. So they enlarge dramatically when B cells are dividing rapidly, producing plasma cells. So when a person is fighting a bacterial infection, they may have enlarged lymph nodes. So again, lymph nodes are going to be the site of adaptive immune response that's going to be mediated by T cells and B cells, as well as accessory cells, which we'll be discussing later. And even though it says there's 3 afferent and 2 efferent vessels, that can depend on the source that you're reading. So the fact that the lymph fluid has to filter through the lymph node before it exits allows the fluid to stay there long enough to allow for inspection of the fluid before it exits. The spleen is about the size of the fist. It's located in the left side of the abdominal cavity, to the left of the stomach and curling around kind of anteriorly. The spleen is served by a large splenic artery and splenic vein that will enter and exit from a concave area of the spleen known as the hilum. And the spleen provides a site for the proliferation of lymphocytes as well as for immune surveillance and response. But even more importantly, the spleen is going to serve as an area for cleansing blood, because this is a site where old or defective red blood cells are going to be broken down by macrophages. And when the parts of red blood cells are broken down by those macrophages, then the parts of the red blood cells can be recycled and used again. They're going to be sent to the liver for that to be done. But, for example, we need to conserve iron for making hemoglobin. So that's one of the functions that occurs in the spleen. Now, it's thought that the spleen may also be a site for erythrocyte production in the fetus, and that normally ends before birth. So within the spleen, we've got a lot of blood vessels, so there's extensive vascularization, and there are basically 2 kinds of tissue that are found in the spleen. It's referred to as red pulp because we've got a lot of erythrocytes and macrophages, and this is where blood has filtered into this organ. And then we have what we call white pulp, and white pulp is where we have germinal centers of lymphocytes. And so lymphocytes are leukocytes. Leukocytes are white blood cells, and that's why it's called white pulp. So when we look at this under the microscope, remember that lymphocytes have a nucleus that's going to take up most of the cell, and it stains dark, so sometimes the white pulp actually appears darker under the microscope. The white pulp tends to cluster around blood vessels in areas known as cuffs, and you can see that in this picture on the right hand side, how there's more white pulp surrounding the blood vessels. And some of that connective tissue will invaginate into the spleen, forming a little trabecular pattern. And again, it's a very, very thin connective tissue capsule. But that's the spleen. We're going to move on to lymphoid nodules. And lymphoid nodules are dense clusters where we find a lot of lymphocytes, and these lymphoid nodules are usually not capsulated, but sometimes you'll find a partial connective tissue capsule. Now, lymphoid nodules are referred to often as mucosa-associated lymphoid tissue, or MALT, and this is found throughout the body. The largest collections of mucosa-associated lymphoid tissue is what makes up tonsils, and special lymphoid nodules in the intestines known as Peyer's patches, and the appendix, as well as throughout the genitourinary, respiratory, and remainder of the digestive tract. Now, there's also in the respiratory, in the bronchi, there's something called bronchus-associated lymphoid tissue, or BALT, and that's usually more commonly found in infants and children. In adults, it's usually not found unless it's needed. So if there's an infection or something that the body needs to fight in the bronchi, then there can be a buildup of bronchus-associated lymphoid tissue in those areas of the respiratory system. And this may not hold true with the text that you have, or it may hold true with the text that you have, but this is the more recent of what most practitioners are finding. Now, if we look at the pictures on this slide, you'll notice at the top picture right in the center of the slide, there is a crypt in the palatine tonsils. The tonsils, and we have palatine tonsils, we have lingual tonsils, we have pharyngeal tonsils, and there's another little group of tonsils known as tubular tonsils. And the tubular tonsils are just a tiny little group of lymphoid tissue that are going to surround the openings for the auditory tube where it goes into the pharynx. Remember, the auditory tube goes from the middle ear to the pharynx. And so those are going to have crypts, which are basically crack-like openings that go deep into the tissue, so that as they're surrounded by the lymphoid nodules, those white blood cells can actually take a closer look at pathogens. When it comes to barrier defenses and the innate immune response, just to act as a reminder, the skin has keratinized cells. Keratin helps make skin tough and more waterproof. And there are also Langerhans or dendritic cells in there in the stratum spinosum of the epithelium that will go and eat up pathogens. And dead cells can actually form a barrier to pathogens. There are different secretions that are produced by our skin, including sweat and sebaceous glands, that produces a lower pH, which makes it more acidic. And then the sweat also provides a washing action. And lipids can be toxic to some pathogens. Our oral cavity has salivary glands that have an enzyme called lysozyme. To lyse means to break or burst apart, so that can break apart pathogens. Our stomach, our gastrointestinal tract, provides a very low pH level, especially in the stomach. We have a lot of acid, so that can kill pathogens. The mucosal epithelium, even though it has non-keratinized epithelial cells, it can trap and facilitate the removal of microbes because mucus is designed to be moved toward the throat where it can be swallowed. So our upper respiratory tract has cilia on the surface of the cells that sweep the mucus towards our throat. The trachea has pseudostratified ciliated columnar epithelium, which sweeps upward towards the throat, so that mucus again can be swallowed and go down to the stomach, where the stomach acid would kill the pathogens. And then we have normal bacteria in the mucosal tissues that will compete for just the surface area of our mucosa, and that can also retard the growth of pathogens. When we look at the phagocytic cells involved in the innate immune response, we see that we've got macrophages, which are agranulocytic cells, because when we look at them under the microscope, there's no little grains. So they are agranulocytes. Those are found in body cavities and organs. They are involved in phagocytosis. They actually have little false foot-like processes known as pseudopodia that will extend around a pathogen to engulf it and bring it into the cell. That's a type of endocytosis. And they also help our lymphocytes. We're going to see how they work in the immune system together. Neutrophils have little grains in them when we look at them under the microscope, so therefore they are a granulocytic cell or granulocytes. Those are found in the blood, and they are also involved in phagocytosis, and they come to the site of infection. They're one of the first types of cells to arrive at the site of infection, because when cells are damaged due to infection, they'll release chemicals, and chemotaxis is that movement of a cell towards different chemicals. They also have and release histamine, which causes increased vasodilation, and in capillaries, when vasodilation occurs, that also causes increased capillary permeability, because if something's going to stretch, it's easier for things to get through the capillary wall. Interestingly enough, histamine in the stomach causes an increase in the production of stomach acid. Now, monocytes are another type of agranulocyte. They're found in the blood, and those are precursors to macrophages and dendritic cells, which we know are phagocytic cells. Natural killer cells are also part of the innate response, and they work by looking for cells that are abnormal, either by having a coating of antibodies on them. Those cells become target cells if they're coated with antibodies, or if they lack proteins that also help determine self from non-self, which are class 1 major histocompatibility complex proteins. It's a mouthful, okay? But those proteins are found on the surfaces of our cells, and they help also determine self from non-self. So if the cell lacks those proteins, saying, hey, I belong to me, then the natural killer cells will go after them, or cells that are engulfed with antibodies, and they'll actually bind to that target cell, and when they bind to it, they perforate it with these proteins called perforins, and they release enzymes called granzymes into the cell, and then the cell just kind of bursts apart. So that's how natural killer cells work. Our natural killer cells, as well as other cells of the innate immune system, our phagocytic cells, also have the ability to recognize the patterns of molecules found on pathogens, such as the components of a bacterial cell wall or the proteins on the flagellum of bacteria. And they use these pattern recognition receptors, which are membrane receptors on the innate cells, to recognize the characteristic features of pathogens and molecules that could be released by stressed or damaged cells. And the receptors on our cells are thought to have evolved prior to adaptive immune responses. And they're present on the surfaces of our phagocytic cells and our cytotoxic natural killer cells, whether we need them or not. However, the variety of these receptors is limited. It's limited because a lot of DNA is required to produce them, and also the surface area of our cells is limited. So the strategy of these receptors is limited and does not really compare to the specificity and response that our adaptive immune response can create. But should these cells come in contact with a species of pathogen that they recognize, the cell can bind to the pathogen and initiate phagocytosis or cellular apoptosis, and that can help destroy the pathogen. If we look at this particular chart, what it shows us on the left side, the far left, is the actual name of the receptor on our cell surface that's going to bind to the ligand on the pathogen, and there are different types of ligands that you see. And the chart also shows the source on the far right side, so it might be a mycobacteria or fungi or virus. And please, this is not a chart that you would need to memorize, so it's just a demonstration. Next, we're going to discuss soluble mediators, and understand that a mediator in terms of physiology is simply a chemical that is going to be involved in the process of a response. And these are soluble mediators, so we know that they are soluble in fluids, in body fluids. We have blood, we have lymphatic fluid, we have interstitial fluid. And so cytokines are chemicals that are released by cells that only work over very short distances, and they can cause the receiving cell to change its physiology, how it functions. Chemokines are the chemicals that attract other cells in the process of chemotaxis over longer distances. Interferons are antimicrobial proteins that help enhance our immune response. They activate macrophages. That word should be activate, not active. They activate macrophages and natural killer cells. They also help interfere with by blocking viral replication in cells that are still somewhat healthy by interfering with protein synthesis, also by degrading viral RNA, or sometimes even causing cells that are infected to undergo apoptosis, which you can see in the picture on this particular slide. And lastly, there are proteins that are known as opsonins that cause opsonization, which is to make tasty, and that means that we're going to tag a pathogen by binding an antibody, which antibodies are proteins, or maybe binding an antimicrobial protein such as mannose-binding lectin. Mannose-binding lectin is a protein that binds to mannose residue produced by bacteria. C-reactive protein is another protein that can bind to infected cells or damaged cells. So those are examples of soluble mediators. The innate immune response also includes a system of chemical reactions known as the complement system. And this system actually belongs to the innate response as well as to the adaptive response. Complement includes a series of 20 different circulating plasma proteins, including things like lectins and opsonins and other regulatory proteins and factors that will become activated during an encounter with a pathogen. And these proteins are produced in the liver. And if we look at this picture, we can see that there are 3 different pathways involved in the complement system. The pathway on the upper left, the classical pathway, is actually part of the adaptive immune response because it's activated by antibodies that coat target cells, and we're going to learn more about the adaptive immune response. And then the lectin pathway and alternative pathway are part of the innate immune response. However, all 3 pathways are going to converge at a specific protein known as the C3 complement 3 protein that continues a cascade of responses that lead to opsonization, where the pathogen is coated, and that enhances phagocytosis. It's going to enhance inflammation, and so we're going to see the release of histamine, which increases blood vessel permeability and also attracts phagocytosis by chemotaxis. And in the middle of this picture, you see the letters MAC and a whole bunch of little proteins that are involved in the production of MAC, which stands for a membrane attack complex. The membrane attack complex is a bunch of complement proteins that are going to fit together to form a pore in the membrane of the target cell. And so when we get pores in the membrane of the target cell, water from outside of the cell can now come in and cause the cell to burst and break apart. So that's what the membrane attack complexes do. They form these damaging pores in the membranes of pathogens. So again, complement system involves both innate and immune responses when it comes to our defense systems. The next part of the innate response that we're going to discuss is inflammation. Now, we learned in A&P1 that areolar connective tissue, which surrounds blood vessels, surrounds lymphatic vessels, and underlies epithelial tissue, not only has fibroblasts that make collagen fibers and reticular fibers and elastic fibers, there are also found within areolar connective tissue other cells called mast cells, as well as macrophages. And when tissue damage occurs because cells are either exposed to too much heat or chemicals or electricity or trauma or bacterial toxins that can damage the cells, when that damage occurs, the mast cells will release inflammatory chemicals, including histamine, which causes vasodilation of the capillaries, which increases capillary permeability. They'll also release heparin, which keeps blood flowing to that area of tissue damage where the cells are damaged in some way, shape, or form. And not only do the mast cells release histamine and heparin, they can also release prostaglandins and leukotrienes. As well as the mast cells, the injured cells themselves, along with local phagocytes and basophils, can also release prostaglandins and leukotrienes. So prostaglandins are important because they'll also relax the smooth muscle in that area where that capillary is, allowing it to vasodilate. And prostaglandins are also important for signaling for pain when there's tissue damage. That's why non-steroidal anti-inflammatory drugs work to inhibit the production of prostaglandins. Leukotrienes are important because they're going to attract neutrophils and other macrophages into the area, and they do that by way of chemotaxis. Leukotrienes can also increase vascular permeability. And after the neutrophils have come to the site, other phagocytes will come to the area, macrophages will come, and they'll continue to clean up the cellular debris. And if a local infection is severe, after the neutrophils and macrophages eat up the pathogens, they die. And when a person has a collection of pus, that's basically just the dead cellular remains of those cells, the neutrophils and the macrophages. So Inflammation comes in acute forms where it's a very short-term response to tissue damage. It can become chronic, so if there's an ongoing presence of a foreign body or a persistent pathogen or an autoimmune disease, that can cause a chronic state of inflammation. With a fever occurs when leukocytes and macrophages are exposed to pathogens, and those pathogens cause the release of chemicals known as pyrogens from the leukocytes and macrophages, and that causes the hypothalamus to increase body temperature. The reason for that is most bacteria prefer normal body temperature. By having the hypothalamus increase body temperature as a response known as a fever, that makes an inhospitable environment for the bacteria, and that can also help to kill off more bacteria. And that's why fevers are a positive feedback mechanism, because it's a response that is going to increase until we get the desired effect of killing off the bacteria, and then it stops. If we think about the difference between the innate immune response and the adaptive immune response, it's kind of like comparing a lowly foot soldier who's always prepared, responding within minutes to protect the body from foreign substances. That innate immune system basically has two barricades to protect the body. And the first line of defense is intact skin and the mucosae. And the second line of defense is if the first line has been penetrated, internal defenses like proteins that are antimicrobial, phagocytes, other cells to inhibit the spread throughout the body. And the hallmark of that second line of defense in the innate immune system is inflammation. When we look at the adaptive immune response, this adaptive immune response is more like the elite fighting force. They've got the high-tech weapons to attack very specific foreign invaders, and it's going to basically be the third line of defense for the body. So it's going to take more time to mount this third line of defense than it does with that initial innate response. So if we look at the adaptive response, we can see that in being more specific, there are primary cells involved of this adaptive response, which include B lymphocytes and T lymphocytes, which have to be exposed to or primed by an initial meeting with a pathogen or a foreign substance. And that helps the cells to recognize and target those identified pathogens or foreign substances to initiate an immune response. And this increased specificity is helped by receptor development. Remember, receptors are important. This is a very specific response. However, it's systemic. It's not restricted or limited to that initial site of infection. There's also memory involved in the adaptive response. The primary portion of the adaptive response is going to be the first and slowest, as the immune system learns to recognize and then mount a response to that new pathogen. The secondary portion of the adaptive response is much faster and much stronger due to immunological memory. And we also have self-recognition involved with the adaptive response, because we have to have the ability to tell what belongs to our body and what doesn't. We have to distinguish self from non-self, self-antigens versus foreign antigens. Because T cells and B cells are the major players in the adaptive immune response, we're going to look at T cells first and understand that both cell types are produced in bone marrow. And the T lymphocytes have to migrate from the bone marrow to the thymus, where they will become mature. And T lymphocytes are important because they're going to direct control of some of our immunological responses, and they can also mediate B cell responses. And T cells, I think, are anywhere between 65 and 85% of all blood-borne circulating lymphocytes. So of all of our lymphocytes, the T cells make up a majority of the portion of lymphocytes. Now, remember, receptors are important, and on our T cells, we have receptors that have two chains. There's an alpha chain and a beta chain, and there's a variable region of those chains that is farthest from the cell, and it varies from receptor to receptor. But then there's a constant region that's closer to the cell. And remember, this is a cell receptor that's embedded in the membrane of the cell, of the T cell. And so the constant region has less variation. All variation of these receptors is going to begin at the level of our DNA, because these are protein receptors. Remember, structure, function, the structure determines the function. And so the variation in these receptors is going to be a result of the differences in genetics from one person to the next. So each T cell has only one receptor and responds to only one antigen. So in the whole cell, each cell has one receptor on the surface of its membrane and responds to only one antigen. Now, antigens can be complete or incomplete. Complete antigens are referred to as being immunogenic because they cause lymphocytes to proliferate and become reactive, where they react with activated lymphocytes and antibodies that have been released by an immunogenic reaction. Haptens are incomplete. So haptens are antigens that are incomplete. They play a role in hypersensitivity. So we have self-antigens which are not antigenic to us because they belong to us, but they would be to others. So there are different areas of the molecule known as an antigen, and we can look at these areas. We have antigenic determinants that consist of an epitope. The epitope is the place where the antigen can bind to a receptor, and those epitopes are usually made out of protein. Sometimes they're carbohydrates. They vary in their complexity. The proteins are much more complex, and they can produce hundreds of chemically different epitopes. And then that area, again, the epitope is the antigenic determinant. It determines what it can bind to in terms of what our receptors are on T cells. And in this picture, you can't see it in every single part, but all those green cells are T cells. There's the first T cell, the second T cell, and the third T cell in the picture. And the epitopes of the antigen are what are binding to those protein receptors on the T cells. So the variable region of the receptor of the T cell is the farthest from the cell. We said it varies from one receptor to another. We already said that the constant region is closest to the cell and there's less variation. We said that all variation begins at the level of DNA because of genetic diversity, and each cell has only one receptor and responds to only one antigen. So this picture is a little bit misleading because we've got cells with different receptors binding to an antigen. Now, T cells are incapable of recognizing free-floating or cell membrane-bound antigens. They only recognize antigens that are presented to them by macrophages, dendritic cells, and B lymphocytes. So those cells, macrophages, dendritic cells, B lymphocytes, are capable of engulfing pathogens, which we see in this picture. We see a microbe being engulfed by a macrophage. And once that happens, the antigens on the surface of the microbe are going to be broken down, and they will be associated or combined with special proteins known as major histocompatibility complex molecules. And it's the major histocompatibility complex molecule, which once associated with the antigen, will then move outward from that cell to present the antigen to a T cell receptor. And if those match, because remember, structure function, the receptor on the T cell has to match the antigen, then those can combine together. The scenario I described earlier was with MHC2 proteins. The major histocompatibility protein complex class 2 are found only on macrophages, dendritic cells, and B lymphocytes. And those deal with antigens from the outside, from external sources. And they bind with CD4 receptors on T cells to activate and differentiate T cells to become helper T cells, and some can become also regulatory T cells. However, there are major histocompatibility class 1 receptor proteins that are found in all cells because viruses can affect any body tissue. And so these are going to deal with endogenous antigens within the body like cancer or viruses, and those bind specifically with CD8 receptors on T cells, and then those T cells will differentiate and be activated to become cytotoxic T cells and memory T cells. Hey, it's Dr. C. I had to redo this video a little bit because I realized I was saying some things that weren't correct, and I always want to make sure you have the correct information. And then I realized that as I was recording this presentation that you just see my head, which is kind of weird. But this is just this one slide that I'm going to correct and then re-upload and yada, yada, yada. So more about T cells. Naive T cells are CD4 or CD8 T cells that have not yet encountered their specific antigen. And a T cell is going to become activated when two things occur. First, it's going to bind to the correct antigen that's presented by a major histocompatibility complex molecule, but it also has to receive a co-stimulatory signal. Activated CD4 T cells, most of them become what we call helper T cells. However, some of them, smaller number, will become regulatory T cells that help to moderate an immune response. The CD8 cells that become activated are going to be referred to as cytotoxic T cells, and those will directly destroy infected cells, but a few of those can become memory T cells. So that's the part that I wanted to redo for this presentation. This chart shows us different classes of antigen-presenting cells. So with the class 1 multiple histocompatibility complex protein, that includes many cell types in our body because all cells have this protein within them to deal with endogenous antigens such as cancer or viruses, because viruses can infect any cells. So these are not phagocytic cells, but when they have this antigen, that will stimulate them to present the antigen on a class 1 major histocompatibility complex, which can bind with a CD8 receptor and a T lymphocyte, and that would stimulate that T lymphocyte to differentiate into a cytotoxic T cell, which will directly kill the cancer cell or the cell that's infected with a virus. So that stimulates cytotoxic T cell, that's the TC immune responses. Now, the class 2 major histocompatibility complex proteins are involved with macrophages, dendritic cells, and B cells. They are all phagocytic, and the macrophages, when they eat them, that stimulates phagocytosis and presentation at the site of infection. With dendritic cells, they eat them up, and then they actually bring the antigens to regional lymph nodes, which they'll interact with T cells at that point. B cells will eat up the antigens, and then they actually work with helper T cells, which in turn will stimulate antibody secretion by plasma B cells. So there's a lot more involved in that, and we'll discuss that.